My research career started really by accident. I mean, most research careers do, I suppose. My first degree was in maths. I took a degree in maths at Warwick University without any idea of going into biology. I actually didn't know any biology. I was very lucky because Barry Cross at what is now the Bateman Institute offered me the job as a project leader at that institute. So straight from PhD, which was in auditory physiology with a background in maths, I suddenly had a lab of my own and was a neuroendocrinologist and had to discover what a hormone was. When you're dropped in a lab on your own and you haven't got the faintest idea of what to do, you have to find a kind of an interesting question. Something that kind of catches your imagination. And really what caught my imagination as such a kind of cool thing was the problem with the milk ejection reflex. There just seemed to be so many really challenging problems that this phenomenon evoked. And firstly, how's the burst generated, how the cells synchronized, and what does this pulsatile signal mean? And so those are the questions that really engaged me at that stage, because they were kind of questions that resonated with me as a mathematician in some ways. So they seemed the kind of appropriate things to follow. Priming in oxytocin cells that we published in Nature back in 2002, the whole motivation for looking for that prime, for believing that a mechanism like that had to exist, was because we actually kind of knew that a mechanism like that was necessary for our model to work, to produce that kind of bursting. And so actually it was the model that drove the experiments that led us to look for and find priming. The oxytocin system started out as being this kind of lovely system with an intriguing problem. It's now become this really important model system in physiology and neuroscience. It's a kind of template for understanding much of the brain, really.